Good morning, students. We will continue our competition number 3.4 and 5, and today's topic is uronic acid pathway. This is the uronic acid pathway given in books, but we will not study this complete uronic acid pathway, but we will study this pathway up to the formation of glucuronic acid only. So here is the uronic acid pathway. First, glucose is converted into glucose 6 phosphate, which is a common step catalyzed by enzyme hexokinase. Now this glucose 6 phosphate undergoes mutation to form glucose 1 phosphate with the help of enzyme mutase. Then glucose 1 phosphate combines with uridine triphosphate UTP nucleotide. Here UTP is hydrolyzed to UDP and get combines with glucose to form UDP glucose, and two phosphate group, one from glucose and one from UTP, are removed in the form of pyrophosphate. The enzyme required for this step is UDP glucose phosphorylase. Next, UDP glucose undergoes oxidation where CH2OH group is converted to COOH group, that is acidic group, to form UDP glucuronic acid. The enzyme is UDP glucose dehydrogenase. The two hydrogen atoms are removed, which are accepted by coenzyme NAD to form NADH. Next, UDP glucuronic acid is converted into D-glucuronic acid by removal of this UDP group with the help of enzyme UDP glucuronidase. So this is the uronic acid pathway you have to draw in the exam whenever it is asked as a short note. Whenever there is a short note on uronic acid pathway, then they will ask about its significance. So significance of uronic acid pathway is it forms glucuronic acids, or it will synthesize the glucuronic acid. Now this glucuronic acid is hydrophilic compound and it is used for conjugation of various compounds. Sometimes a 2-mark question is also asked, in which they will ask name any 4 compounds which undergoes conjugation with glucuronic acids. So glucuronic acid is hydrophilic compound, just I have told you, which is used to convert the hydrophobic compounds into hydrophilic or amphipathic compounds. This is essential because hydrophobic compounds cannot be transported through the blood and hence they cannot be excreted through the kidney or they cannot be excreted through the bile. Therefore, the hydrophobic compounds must be converted into hydrophilic compounds by combining some hydrophilic moiety, and in this case glucuronic acid acts as a conjugating agent which combines with unconjugated bilirubin, which is hydrophobic in nature, and after combining with glucuronic acid the bilirubin becomes hydrophilic and can be easily transported through the bile and excreted through the stools. Similarly, the steroids are also hydrophobic in nature. They cannot be easily excreted out of the body because of their hydrophobic nature. Hence, hydrophilic compound like glucuronic acid combines with these steroids and make them hydrophilic and excretable form. Similarly, for conjugation of drugs also glucuronic acid is used. Glucuronic acid is also used for synthesis of glycosaminoglycans, which are called as mucopolysaccharides, and as already we have studied in chemistry, mucopolysaccharides consists of one molecule of uronic acid and one molecule of amino sugar. So the main uronic acid present in majority of the mucopolysaccharide is glucuronic acid. So these are the uses of glucuronic acid. Now, effect of drug on uronic acid pathway. The drugs like barbiturates, antipyrine and aminopyrine, when they are taken, they stimulate the uronic acid pathway and therefore synthesis of glucuronic acid increases in the body, and they are used for conjugation of various substances. In some lower animals, vitamin C is synthesized with the help of uronic acid pathway. But in mammals, this step, the conversion of L-gulonolactone into ascorbic acid, does not take place because of absence of enzyme gulonolactone oxidase. This may be asked in MCQ. Why mammals does not synthesize ascorbic acid? So the reason is the enzyme gulonolactone oxidase is absent in mammals, therefore vitamin C is not synthesized. Now we will see the actual step where gulonolactone oxidase is utilized. If you see from this pathway, L-gulonic acid is converted into L-gulonolactone, which is converted into 2-keto-L-gulonolactone, and then which is converted into vitamin C. So the enzyme which is required for conversion of L-gulonolactone to ascorbic acid is gulonolactone oxidase, which is absent in mammals, and hence we cannot synthesize vitamin C in our body. The disease related to uronic acid pathway for us is essential pentosuria. Now this essential pentosuria is one of the member of Gerhardt tetrad disease. Now this Gerhardt tetrad disease consists of 4 diseases: albinism, alkaptonuria, cystinuria and pentosuria. So pentosuria is one of the disease of Gerhardt's tetrad. Now the incidence of essential pentosuria given in some books is 1 in 2500 live births. It is an inborn error of metabolism. The enzyme deficient in essential pentosuria is xylitol dehydrogenase and xylulose reductase. In this case, L-xylulose, which is a pentose sugar, is excreted into the urine, and hence it is called as pentosuria. Now this is the reaction. Here L-xylulose is converted into xylitol and xylitol is converted into D-xylulose. So these two steps requires two enzymes. One is xylitol dehydrogenase and another is xylulose reductase. So due to deficiency of these two enzymes, L-xylulose is not converted into D-xylulose. Therefore L-xylulose is not utilized. This D-xylulose is utilized in HMP shunt pathway, but L-xylulose is not utilized, and hence it is excreted into the urine, leading to essential pentosuria. Again we will come to the main pathway given in the book, where it shows conversion of L-xylulose to D-xylulose. So basically L-xylulose is formed from 3-keto-L-gulonolactone, which is formed from L-gulonic acid, and this L-xylulose is converted to xylitol, xylitol is converted to D-xylulose, and then D-xylulose enters into the HMP shunt pathway or pentose phosphate pathway. So due to deficiency of these two enzymes, L-xylulose is not converted into D-xylulose and excreted into the urine, leading to essential pentosuria. Sometimes it is asked in MCQ. So essential pentosuria is detected by positive Benedict test. Sometimes if the drugs like barbiturate and aminopyrine are given to the patient, they also causes excretion of xylulose sugars into the patient. This essential pentosuria is not harmful to the body, but it has to be distinguished from diabetes mellitus. because when we get Benedict test positive, then we think about the excretion of glucose in the urine, and we will misdiagnose such patient as diabetes mellitus patient. Therefore, this condition has to be ruled out from diabetes mellitus. So this is about uronic acid pathway and its significance. Thank you very much.